Okay, so dear participants, my name is Victoria Gutierrez, and it's my pleasure to welcome you to the second webinar on the EuroBloodNet Topic on Focus webinar program on thrombotic microangiopathies. This program has been accredited with 11 continual medical education credits by the European Board of Accreditation in Hematology. In case that you are interested to apply for these 11 credits, please remember that you have to attend to all the webinars of this program and introduce your name and surname when entering into the Zoom system. Also, please remember to answer the survey that you will receive at the end of the webinar. And also, I will show you later the next webinar of the program and how to register properly. So, for today's lecture is focused on animal models of thrombotic thrombocytopenic purpura, and the lecture will be provided by Karen Vanhoorelbeke. So let me introduce to Karen Vanhoorelbeke. She's a professor at the KU Leuven in Belgium, where she's principal investigator at the Laboratory for Thrombosis Research and chairman at the Interdisciplinary Research Facility. In 2010, she worked at the lab of the professor Tim Sprinkler at Harvard Medical School as a visiting professor. Her research focuses on the molecular mechanism with the hemostatic proteins Van Willebrand factor and its enzyme ADAMTS13 contribute to health and disease. Her group performs fundamental research to understand the role of the Van Willebrand factor and ADAMTS13 in hemostasis, bleeding, and thrombosis, and to unravel their mode of action. These findings form the basis for the translational research lines, where preclinical animal models are established to study disease pathology and new treatment strategies. Her research also contributes in international research projects. Recently, she was awarded with the Horizon 2020 Grant Profile, of which she is coordinator. She is author and co-author of more than 160 publications and a promoter of 30 PhD students. So with this, please, Karen, the floor is yours.
Okay, thank you very much for this introduction. And I would also like to thank the organizers for inviting me to join these webinars on thrombotic microangiopathies. So today I will discuss animal models of thrombotic thrombocytopenic purpura. Here are my conflicts of interest. So what are the learning objectives today? So I have two main parts in my talk. The first one is focused on the different types of animal models that have been developed for TTP. What are the signs and symptoms that reflect TTP in humans in these animal models? And are there triggers needed to induce TTP in these animals? And then the second part of my talk will focus on the use of these animal models in drug discovery. So first, a brief introduction of thrombotic thrombocytopenic purpura. I'm sure you all know this, but just to highlight what do we see in TTP and what do we expect in an animal model. So TTP is a rare disease or an ultra-rare disease. It is a devastating disease because patients end up in intensive care when they have a TTP boot. The disease is lethal when left untreated, and it is a chronic disease. So patients suffer lifelong from TTP as there is no cure for TTP. There are two types of TTP. About 5% of the patients, so the minority of the patients, suffer from congenital TTP due to mutations in their ADAMTS13 gene. And the majority of the patients, about 95%, suffer from the immune-mediated form of the disease, and that is caused by the development of autoantibodies, as depicted here by this figure on the slide. So it is caused by autoantibodies against ADAMTS13 in these patients. So now what causes TTP? TTP is caused by a defect in the VWF cleaving protease ADAMTS13. Here we look at a diagram of a blood vessel in a healthy individual. And you can see that the endothelial cells line the vessel wall in our circulation. And these endothelial cells, they produce and secrete the hemostatic protein von Willebrand factor, or VWF. And this protein is a multimeric protein. When you analyze it on an agarose gel, you see the dimers, the tetramers, the heptamers, and the octamers, etc. So it is secreted as an ultra-large protein, meaning that you have multimers of an ultra-large size, and their size can be up to 20 million Dalton. And in this way, VWF is the largest multimer found in our blood. Now, these ultra-large VWF multimers, they are protothrombotic, they are dangerous, and therefore they are cleaved by the VWF cleaving protease ADAMTS13 when they enfold into the flowing blood, and into multimers of a lower size, but still a size that is big, and we call it the high molecular weight VWF multimers. If we look what is happening in a TTP patient, we see that the VWF cleaving protease, here presented as a scissor, is deficient in TTP. So it does not work, meaning that the ultra-large VWF multimers are not cleaved, and they can spontaneously bind to platelets, and microthrombi are formed. And these microthrombi, they block the arterioles and the capillaries in many organs. So what are the signs and symptoms of TTP? Because of the formation of the microthrombi, the platelets are consumed in these microthrombi, and patients suffer from severe thrombocytopenia. Because the red blood cells are pushed through these microthrombi, patients suffer from hemolytic anemia. The red blood cells rupture, and you can see them here in a blood smear. I hope you can see my cursor actually, as these little moons that you can observe where the arrows are in the blood smear. They have microthrombi, but in many organs like the heart, the kidney, and the brain. And here you see tissue sections, unfortunately, died because of a TTP episode. And you see the microthrombi were stained for VWF, and you see the VWF staining in these microthrombi. And because of the microthrombi, there is organ damage and organ failure. Now, we know that patients with congenital TTP can have a TTP boot when they are born, but we also know that patients can live for years with an ADAMTS13 deficiency, a severe deficiency, and they do not show any signs or symptoms of TTP. And that is because there is an external trigger needed to induce TTP, and the known triggers are pregnancy, stress, surgery, infection. But what the actual biochemical component is that induces this TTP episode, we still do not know. So animal models for TTP, what would be an ideal animal model? Of course, we need two types of animal models because we have a congenital form of TTP and we have the immune-mediated form of TTP. If we want an animal model for TTP, obviously ADAMTS13 must be deficient. That is here represented as a broken scissor, which I will use throughout my talk. And if ADAMTS13 is not working, then I just explained that you will find ultra-large VWF multimers in TTP animal models, like you find in TTP patients. The animal should have the signs and symptoms of TTP, meaning the severe thrombocytopenia, the hemolytic anemia, the microthrombi in the different organs, and markers for organ failure like lactate dehydrogenase or LDH, that levels that are increased. And of course, the question will be, will we need a trigger to induce TTP in animals? So let's zoom in on an animal models for congenital TTP. So there are two models: mouse model that was developed in 2005 by the group of Dave Ginsburg in the USA, and the zebrafish model that was developed in 2020 by the group of Long Zheng, also in the USA. I want to give you some history on the mouse models for TTP because there were some interesting findings. So the first mouse model made by the group of Dave Ginsburg was made on a genetic background. The numbers might sound complicated, but it means it was on a black 6 background and a 129 SV background. And the black 6 mice, or mice with genetic backgrounds that are often used in research. So they were a logic choice. They disrupted the ADAMTS3 gene by standard technologies, and they confirmed that the knockout mice they generated did not have any ADAMTS13 activity. As you can see here in the Western blot, in the wild type and the heterozygous mice, you see a band appearing in the Western blot, meaning that ADAMTS13 can cleave the VWF substrate, while in the knockout mouse, you see only the original band of the VWF substrate and no band appearing. So ADAMTS13 is deficient in these mice. So they are knockout mice. What we would also expect if they are knockout mice, that that the ultra-large VWF multimers would appear, and they are situated here in the gel. But intriguingly, also the wild-type mice had ultra-large multimers, which was rather unexpected. Now, did the mice develop TTP? No, there were no spontaneous TTP signs and symptoms. There was a normal development of the mice and survival, which was also unexpected. And importantly, pregnancy did not trigger TTP in the mice. So we also have the mouse colony in our lab, and the mice breed perfectly well. Pregnancy is not an inducer of TTP in the mice, in contrast to what we see in humans. Now, this was very intriguing. Why do we not see TTP in these mice? So the researchers reasoned that since ADAMTS13 and VWF are closely linked in the disease, what if they would increase the VWF levels in these mice? And they could do that by backcrossing the mouse to the KSA-RK strain, that is a strain with 5 to 10-fold increased VWF levels. Of course, the mice were still deficient in ADAMTS13. And now you see that the ultra-large VWF multimers present in the knockout mice were absent as it should be in the wild-type mice. But again, normal development and survival in the mice. No, pregnancy did not induce TTP, but some sporadic mice spontaneously developed TTP signs and symptoms. But this was not statistically significant. So we cannot say that we have TTP in these mice. So now they looked for a trigger to induce TTP. And the trigger they focused on was Shiga toxin. And Shiga toxin is a toxin known to induce the thrombotic microangiopathy, hemolytic uremic syndrome, or HUS. The Shiga toxin consists of A subunit and a B subunit. And when it binds to endothelial cells, like shown here, it is internalized and it goes via the Hoagie to the endoplasmic reticulum. And there the A subunit, which is a toxic subunit, blocks the ribosomes. And if the ribosomes are blocked, there is no more production of proteins, so no more translation of the messenger RNA, and the cells die, of course. Now, it was shown that the Shiga toxin B subunit, so the B subunit, as you can see here, which is not the toxic part, but that the B subunit, when it binds to endothelial cells, it actually increases the release of ultra-large VWF multimers. And here on the left photograph, you can see these ultra-large VWF strings. So the researchers reasoned if we will treat the mice with Shiga toxin, we might induce TTP. So they injected the mice with the Shiga toxin, the mice on the black 6 background, there was no TTP. But if they injected the mice on the background with the higher VWF levels, they could induce TTP, but not in all animals. So why do they have TTP? Because they have severe thrombocytopenia in 5 of the 13 mice. So here you see with the different colors, the different mice, the platelet count, and you see that in the mice here, you have severe thrombocytopenia, while in the other mice, the platelet counts were higher or even did not change. And also in some mice, you see a decrease in hemoglobin, reflecting the hemolytic anemia, and you see in the knockout mice, in some of these mice, the schistocytes appearing, also reflecting the hemolytic anemia. So some mice developed TTP. They also had microthrombi, but the microthrombi that you find in these mice, there are few, but you find them in different organs, in the brain, the heart, the kidney. And in the middle panel, they stain the microthrombi for VWF. So you see they are rich in VWF. And in the right 3 panels, they stain them for fibrin. And there is no color for fibrin because it should be red. So, and this is typical for TTP, of course, that you have VWF-rich microthrombi. So Zika toxin could induce TTP in a subset of mice. Now, Zika toxin is one trigger. The team of Hans-Peter Rottensteiner and Friedrich Scheiflinger from Baxalta, the company Baxalta at that time, which is now taken over by Takeda, they developed recombinant VWF for the treatment of patients with the bleeding disorder von Willebrand's disease. And that team reasons that what if we would inject extra recombinant VWF that contains ultra-large VWF? They knew that extra ultra-large VWF will probably spontaneously bind the platelets and induce microthrombus formation, which would then again lead to the signs and symptoms of TTP. So they did that. They injected this recombinant VWF in the black 6 mice, so the mice that were not susceptible to TTP when induced with when triggered with Zika toxin. When they injected those mice with these large amounts of recombinant VWF that contained ultra-large VWF, they saw that they had severe thrombocytopenia in all mice tested. So you see the platelet count in the untreated mice, normal platelet count. Counts when injected with recombinant VWF, you induce severe thrombocytopenia, which is a sign and symptom of TTP. You also induce hemolytic anemia. There is a decrease in hematocrit and some schistocytes appear. And they did find organ damage, but mainly in the heart. They found macroscopic lesions in the heart, some platelet VWF aggregates, which I point out here, but are difficult to see. Here, a VWF-rich aggregate, and also increased lactate dehydrogenase levels, which point to the rupture of red blood cells and the organ damage. So recombinant VWF does trigger TTP in ADAMTS13 knockout mice. Let's move on to the zebrafish model. Zebrafish model is also an interesting model. The fish can be breed easily, and it's a good model to make a knockout zebrafish. So what did they do? They used the CRISPR-Cas technology to disrupt the ADAMTS13 gene. And when they looked in Western blot, they found no expression of the ADAMTS13 protein, and also there was no ADAMTS13 activity, represented here again by the broken scissor. They checked for the presence of ultra-large VWF multimers, because if ADAMTS13 is deficient, we should find how high more ultra-large VWF multimers in the knockout zebrafish, here in lane 4, 5, and 6, and less in the wild-type zebrafish in lane 1, 2, and 3. You can already clearly see that the multimer pattern in the zebrafish looks different than what we see in mice, and that the difference in high molecular weight multimers, and especially the ultra-large multimers, it's there, but it is more difficult to see. It's just another pattern. Also, the zebrafish do not develop spontaneous TTP. They saw some mild thrombocytopenia, as you can see here in the figure, a small decrease in platelet count, some mild hemolytic anemia, slight increase in ruptured red blood cells or fragmented red blood cells and schistocytes, as you can also see here in the blood smear, but also no spontaneous TTP. Yeah, the zebrafish model was developed by the group of Xinglong Zheng in the US, as I said, and they reasoned that maybe a trigger for TTP could be histones. Just a reminder, a histone is a protein found in the cell nucleus that is highly positively charged and that is present to wound the DNA, so that the DNA that is 1.2 meter actually can be compressed in a chromosome which then fits in a cell nucleus. The work of Vanhoorelbeke showed that patients with the TMA and TTP have increased levels of histones in their blood. So histones could be a good trigger for TTP. And indeed, when they treated the zebrafish with these histones, they saw a more profound thrombocytopenia in the knockout zebrafish, here depicted at day 14, compared to wild-type zebrafish. Also, there was a mild hemolytic anemia. The figure here represents the difference in erythrocytes in zebrafish, in knockout zebrafish at day 0, 1, and 7. And there was a decreased survival. So we also know that when patients are not treated when they have an acute TTP episode, the mortality rate is really high. You might have noticed that also in the mice, what I just discussed before, the mice did not die with the shigatoxin levels that were injected. But here in the zebrafish, they saw that the zebrafish, when TTP was induced, they did the frequency of death in this zebrafish was higher. Here in red is the curve where we have the knockout zebrafish treated with the histones, compared to the wild-type zebrafish treated with the histones in purple, or wild-type and knockout zebrafish treated with PBS in green. They also looked for microthrombi, and they found them in the large microvascular thrombi in the liver, and in the mesenteric vessels of these knockout zebrafish after induction with the histones. And here you see a staining of the tissue for VWF, and you see that the microthrombi are again positive for VWF, which is typical for a TTP microthrombus. So what can we conclude from the animal models for congenital TTP is that ADAMTS13 deficiency is not lethal and it is well tolerated in these models. This can reflect the situation in some patients where patients can live for a very long time without a TTP boot, even if ADAMTS13 deficiency is there. The signs and symptoms we pick up, but to various extents. The thrombocytopenia can be very severe or only moderately decreased, or not in all animals decreased. There is hemolytic anemia. There are microthrombi, but they are sometimes difficult to find since we have the mouse model also in our lab. But there's always organ damage, which you can see in an increase in LDH. The triggers needed to induce TTP in these animal models were shigatoxin, recombinant VWF, or the lysine-rich histones. And these triggers probably stimulate ultra-large VWF release. Well, probably they do stimulate ultra-large VWF release, but probably these ultra-large increases in ultra-large VWF help to set in TTP episodes, but other genetic factors will probably also play a role, like variations in platelet vessel wall function. And the triggers in humans I have discussed before. Then we go to the animal models for immune-mediated TTP. And what are these? I'm not going to discuss all of them, but I want to highlight some of them and highlight some important characteristics of these animal models. So we again have a mouse model, we have a rat model, and we have a baboon model. So now if we want to develop a mouse model for immune-mediated TTP, the idea would be that the mouse itself, or the baboon itself, develop an immune response, or develop an autoimmune response against their ADAMTS13, and that we in this way mimic the situation in patients. However, this is very difficult. So therefore, a logic thing to do was to inject the wild type animals. So now we work with the wild type animals, in this case the wild type mice, with an antibody that inhibits ADAMTS13 activity to create ADAMTS13 deficiency in these mice. Our group did that by developing murine, so murine monoclonal antibodies against the human ADAMTS13 that inhibit ADAMTS13 function, and a group of Long Zheng cloned a human antibody from a TTP patient that also inhibited ADAMTS13 activity. I depicted here the ADAMTS13 molecule, as you can see, that consists of multiple domains, and in the red domain you find, or this is the metalloproteinase domain, you find the catalytic site where that ADAMTS13 uses to cleave VWF in the A2 domain. So at the bottom of the figure you see the part of the VWF subunits, and you see that the A2 domain contains the cleavage site. So it is known that ADAMTS13 binds via its spacer, cysteine, disintegrin, and metalloproteinase domain to the unfolded A2 domain, and then cleaves VWF. So if we use antibodies, I have to that's my next slide. So if you use antibodies that block one of these interactions or block the active site in the metalloproteinase domain, you can make ADAMTS13 deficient in these animal models. So I will show you our model. So we developed a large set of murine antibodies, murine ADAMTS13 antibodies, and against the first part of ADAMTS13, and two of them inhibited ADAMTS13 activity. So when we inject them in the mice, you see this is the ADAMTS13 activity in normal murine plasma, or NMP. And when you combine both antibodies, you see in the bottom curve, the black dots, that you have full inhibition of ADAMTS13 activity. So we know that the antibodies are directed against the first part of ADAMTS13 and that they completely inhibit ADAMTS13. If ADAMTS13 is deficient, we should see the ultra-large VWF appear, and that is what we do. So in this experiment, we injected wild-type mice with the control antibody, 2810, so the open circles, and then wild-type mice with the inhibitory antibodies, the black circles. So you see that before injection of the antibodies, the multimers are of course the same, but once you inject the antibodies at day 1 and day 2, you see that we see an increase in percentage of high molecular weight multimers when we inhibit ADAMTS13 function, the closed circles, but not when we inject the control antibody, the open circles. And you can see that on the multimer shell. I will just pick out 2 lanes you have to focus at. Look at day 1. If you inject the control antibody 2810, you see the normal multimer pattern. If you inject the 2 inhibiting antibodies, you see an increase in these high molecular weight multimers, which also contain the ultra-large VWF multimers. But the mice did not develop spontaneous TTP, just like in the congenital TTP, and therefore we used recombinant VWF as a trigger. And as you can see, when we did that, we could induce severe thrombocytopenia in all mice. If you look at the graph, you see the platelet counts in function of time, and with the black circles, when we inject the inhibitory antibody, so when ADAMTS13 is deficient, we have severe thrombocytopenia, and not when we inject the control antibody. We also observed hemolytic anemia and organ damage, which we measured by an increase in LDH. So again, the black dots 24 hours after injection of recombinant VWF and the inhibitory antibodies, you see an increase in LDH, which is not the case with the control antibody. And then I would like to end with the baboon model of TTP, which we developed. So when the mouse model for TTP was published, and we saw that, so the mouse model for congenital TTP, and we saw that there was no spontaneous TTP, also not when the mice were pregnant, we thought of developing another animal model and looked to the baboons, because baboons, of course, are closer to humans. And we thought, okay, let's see what this gives. Of course, we cannot make a knockout baboon. So the only thing we could do is we could make an immune-mediated model, an immune-mediated TTP model in baboons. And how did we do that? We did that by injecting in a baboon a monoclonal antibody that is directed against the active site in the metalloprotease domain. So when you do that, you block the active site of the enzyme. The enzyme is deficient. And we do the first step. We actually have an immune-mediated TTP model. So we injected in the baboons 2 times the antibody. These are the black arrows, day 0 and day 48. We took blood samples and we followed, of course, the activity of 8-MTS13. And if we block 8-MTS13 in baboon, you here see in the curve a nice inhibition of 8-MTS13 activity. And the squares represent injection of a control antibody in the baboon, and you see no inhibition of 8-MTS13 activity. Inhibition of 8-MTS13 activity clearly resulted in an increase of ultra-large multimers, as you can see in lane 1 and lane 2, which correspond to day 1 and day 2 after injection of the inhibitory antibody. Now, what with TTP? We were actually very surprised and also happy to see that there was spontaneous TTP in all baboons, because without injecting any trigger, the baboons developed spontaneous TTP signs and symptoms when we simply blocked 8-MTS13 activity. You can see here a decrease in platelet counts, the circles, when we inhibit 8-MTS13 activity, the squares, no decrease in platelet count when we use a control antibody. We saw hemolytic anemia, a decrease in haptoglobin when we injected the inhibitory antibody, not when we injected a control antibody. And the same, a decrease in hemoglobin and an appearance of the schistocytes, 3 markers of hemolytic anemia. We also saw an increase in LDH, which reflects damage of red blood cells and organ damage. Again, the circles when we inject the inhibitory antibody and not with the control antibody. And importantly, we even saw the appearance of microthrombi in different organs of the baboons. So here you see the kidney, the heart, the brain, the lung, and the spleen. 3H9 is the antibody that inhibits 8-MTS13 activity. 5C11 is the control antibody. So you see, if we stain the microthrombi for platelets, which is CD61, you see this brown color appearing in the heart, the brain, the spleen, but not the lung. And you do not see these microthrombi in the control baboons. If we stain the microthrombi for VWF, you see the brownish color. So the thrombi are platelet VWF rich, which are typical for TTP microthrombi. And they were negative for fibrin, which is the MSB staining. And if they would be positive for fibrin, you would expect a red color. So that was very exciting. The baboons developed spontaneous TTP. But we have to say these are the early stages of TTP, because when you look to the baboons, they did not look really ill. They looked quite okay. None of the baboons died, so they had the sign and symptoms of early stages of TTP. So what can we conclude? We can make immune-mediated TTP animal models in mice, rat, and baboons by injecting antibodies that inhibit 8-MTS13 activity. So by making 8-MTS13 deficient using antibodies. In the mice, it's mouse or human anti-MDTCS or anti-S antibodies. In rat, I did not discuss that, but that model will come back. We use polyclonal anti-human antibodies, anti-human anti-8-MTS13 antibodies, I should write. And in the baboon, we use this mouse anti-metalloprotease domain antibody. Mice and rats, ADAMTS13 deficiency is not lethal and is well tolerated, like in the knockout mouse. But in the baboon, simple ADAMTS13 deficiency leads to the early stages of TTP. So this was actually the proof that ADAMTS13 deficiency indeed is linked to TTP. The signs and symptoms in the immune-mediated TTP model in the mice and the rats is again to various extent, and in the baboons it is clear. All baboons have clear signs and symptoms of TTP. In mice and rats we need triggers, but in baboons we do not need triggers for the early phase of TTP. But of course for end-stage TTP we might also need a trigger. So now I come to the last part of my talk, and that is how were these animal models used to develop novel therapies for TTP. I will zoom into two therapies. Other therapies have been tested, but I cannot discuss them all here in my talk. And the first therapy is recombinant ADAMTS13. Apologies. I see something is missing on my slide, but okay, I will explain. So you know probably that TTP patients are either treated with plasma infusion or plasma exchange to replenish the defective ADAMTS13 by an active ADAMTS13 from a healthy donor. And you all know that giving plasma to patients is cumbersome for the patient and can also be dangerous. So again, Baxalta, now Takeda, developed recombinant ADAMTS13 as a novel therapy for TTP. So they wanted to check if you simply inject the protein itself without any plasma component, can you treat TTP? And the first thing they tested was, can it be used as a treatment for congenital TTP? So they used the knockout mouse model and checked if we induce TTP, but we give recombinant ADAMTS13 before actually TTP starts or sets in. Can we prevent TTP? So therefore, they took a blood sample. Before the experiment, they injected saline in the mouse, recombinant VWF to trigger TTP, and another blood sample. And what they see, as I just explained, severe thrombocytopenia, hemolytic anemia, and organ damage in these mice. But if they injected, instead of saline, recombinant ADAMTS13, then you see that the mice did not develop TTP. So recombinant ADAMTS13 can be used to prevent TTP in congenital TTP mouse models. So no plasma is used here. Then our group, together with Baxalta, checked whether it can also, recombinant ADAMTS13 can also be used to treat immune-mediated TTP. One can think, okay, if we have immune-mediated TTP, there are so many autoantibodies present in the patients that if we inject ADAMTS13, these autoantibodies will probably immediately kill the ADAMTS13 that injected, and it is of no use. So therefore, we tested this in the rat model of immune-mediated TTP. So how did the experiment go? Again, a blood sampling before the start of the experiment. We injected the rat mouse with the polyclonal anti-human ADAMTS13 antibodies. And we know that in this way, we block the endogenous ADAMTS13 from the rat, but also we will block ADAMTS13 that will be injected. So you either inject saline or recombinant ADAMTS13 and then trigger the TTP in the rats by injection of recombinant VWF, and then other blood samplings. And what do we see? So we see, look at saline, so the bottom curve, you see the severe thrombocytopenia, so the platelet counts when you simply inject saline and you trigger TTP. But when you inject different doses of recombinant ADAMTS13, especially the 1,600 units per kilogram, so before you trigger the rats with recombinant VWF, you see that the platelet counts do not change. So you can prevent also immune-mediated TTP in the rats. And also, if you have TTP when you inject saline, you have a reduction in hemoglobin, but the highest level of recombinant ADAMTS13 injected in the rats prevented the hemolytic anemia. So this means that the recombinant ADAMTS13, or the amount of recombinant ADAMTS13 you inject, actually overrides the large amount of inhibitory clearing autoantibodies that are present in these rats. You can see here that recombinant ADAMTS13 that is injected, you see the square, open square, that forms immune complexes. Here you see the immune complex titer. It forms indeed immune complexes with antibodies that were injected in the rat, as expected. But if you look at the right-hand graph, you see that with the high amounts of ADAMTS13 we inject, there is still enough ADAMTS13 present to have a high ADAMTS13 activity in these rats, and that explains why the rats do not develop TTP. And then the last novel therapy I want to discuss is actually something completely different. So our group and other groups have been studying the interaction between ultra-large VWF and the platelets already for a long time, and developed monoclonal antibodies that could block this interaction and could serve as good antithrombotic agents. Now, our team developed an antibody against the platelet receptor that is responsible for the interaction with VWF. And then years later, also Uplinks, now Sanofi, developed a nanobody that recognized the A1 domain in VWF and also blocked the interaction with the platelet. And that nanobody is the antibody you all know well, caplacizumab. So we reasoned so many years ago that maybe we can, although we do not replace the defective ADAMTS13, maybe we can treat TTP patients if we block microthrombi formation. And this can also be very valuable because these microthrombi block the capillaries and they cause organ damage. So maybe this therapy could be good. That was our initial reasoning. If there is no plasma exchange readily available, you inject or you block platelet von Willebrand factor interaction, you prevent formation of microthrombi, and that would help the patient in the end. I have to say, remember when we discussed that therapy and planned our experiments in baboons, there was a lot of criticism because TTP patients already have a severe thrombocytopenia. So if you then would extra block platelet VWF interaction, that might not be good. But anyway, science needs new discoveries. So we did do the experiment in the baboon. So as I explained, we took baboons again and we injected them with the antibody that blocked ADAMTS13 function. So we created deficient ADAMTS13 in the baboons, but now not only for 2 days, but for up to 10 days. And we first checked, can we prevent TTP? So can we, when we inject, you see the black triangle. So when we inject the antibody to block ADAMTS13, and then immediately thereafter inject the antibody that prevents microthrombus formation, can we prevent TTP? So we have the control baboons, we inject the antibody to create ADAMTS13 deficiency, and as expected, we have TTP signs and symptoms, severe thrombocytopenia, hemolytic anemia. And this graph shows you the interaction between the VWF and the platelet, and you see that this is quite constant. Of course, there is no inhibition of interaction between the VWF and the platelet because we did not inject any extra antibody. But now, in the prevention group, we injected the antibody to make the baboons deficient in ADAMTS13, and immediately thereafter, we injected the antibody that prevents microthrombus formation. And you clearly can see that there is no TTP, no thrombocytopenia, a decreased hemolytic anemia, and you clearly see that indeed the VWF platelet interaction is fully blocked. But then what about treatment? So we took, can we also treat TTP patients? because that is the most logic thing to do. Patients end up in the hospital with the TTP boot, so you need to treat them. So we have had, of course, the same control group. And now what was the treatment group? In the treatment group, we first injected the antibody, 3H9. And only after day 4, when we know the baboons had TTP, we injected them with the antibody that prevented formation of microthrombi. And that for up to day 10. So the control group, of course, the same data. But if we now look in the treatment group, you see, and injected 2 antibodies, you see that when we inject the inhibitory antibody, TTP appears. But when we have TTP and then start to inject the antibody that prevents microthrombi formation, you see the platelet count increasing. So that was a nice proof that we can treat TTP by blocking actually platelet VWF binding interaction. The hemolytic anemia only recovered slowly. And here again, you see when there is no platelet VWF inhibitor injected, the VWF activity is the same. But as soon as you inject the inhibitory antibody, so the antibody that blocks platelet VWF interaction, there is a decrease in VWF activity. So what can we conclude from the novel therapies? Recombinant ADAMTS13 is an effective agent to treat congenital and immune-mediated TTP in mice and rat models. Clinical trials are ongoing with for congenital TTP patients and are very successful, and they also started now for patients suffering from immune-mediated TTP. Anti-VWF and anti-GP1B antibodies that inhibit the microthrombi formation are also very valuable, as we and then later Uplink showed that they are very effective in treating immune-mediated TTP in the baboon model. And yeah, this is clearly reflected by the success of caplacizumab treatment of ITTP patients. So what are my take-home messages? That animal models of TTP represent the main characteristics of human disease, that they aid in understanding the pathophysiology of TTP, and that they have been and are very valuable in drug discovery. And with this, I would like to thank our team of the Laboratory for Thrombosis Research, and especially Dr. Louis de Forge, who developed a mouse model of TTP, Professor Hendrik Vees, at that time postdoc in our lab, who developed a baboon model for TTP, and Professor Claudia ter Steeg, who is here on the picture, who is the new PI in our group, who worked on the recombinant ADAMTS13 as a treatment for immune-mediated TTP in the rat model. I also would like our collaborators on this project, the people from Baxalta, the people from the University of Free State in Bloemfontein, South Africa, and Glenmark Pharmaceuticals for the anti-GP1B antibody, and also the funding agencies. And with that, I am ready to take questions.
So thank you very much, Karen, for this lecture. In the meantime, Karen, I have noted a couple of questions for you that maybe you can answer. And if any of you have any further question, write it on the chat to be answered. The first question is, what is your opinion, the best animal model for TTP?
The best animal model of TTP, it depends a bit on how you look at it, I would say. If I say what is the best animal model, for example, to test novel drugs, then I would say the baboon model, for several reasons, because baboon is closer to humans than mice and rats, because in the baboons, if you only have to inject an antibody that inhibits ADAMTS13, no trigger needed, and always all baboons develop TTP, which is important if you want to check the reproducibility of your results. And also you can take more blood from these large animals which is very important because you have to measure a lot of parameters in this model. If you have to get it with mice, you need to use more mice to be able to take enough blood. If the baboon model represents the human disease the best, that's of course another question, because the baboons develop spontaneously the early stages of TTP. So, okay.
Okay, thank you very much. There is a question on the chat.
Mm-hmm.
Karin, do you want to proceed?
Yes. Now I have to see. Johanna asked a question for the mouse model, but it's not. There it is. Do human anti-ADAMTS13 antibodies from ITTP patients inhibit mouse ADAMTS13 and induce an open conformation? I know that the cloned human ADAMTS13 antibody developed by Xinglong Zheng cross-reacts with murine ADAMTS13, so he could develop a mouse model. I know that the one developed by Vanhoorelbeke does not. So we really have to, if we isolate antibodies from humans, we have to check that and see if they cross-react, yes or no. Do they open an ADAMTS13 conformation? If we work with ADAMTS13 in the black 6 mice, we cannot induce an open conformation because they have the short form of ADAMTS13, and also some work of Evan Sadler showed that allosteric activation of mouse ADAMTS13 is not really obvious. So that is for Johanna. Then I have Mario. I'm sorry, I don't think I can pronounce your last name properly. Made differences in severity of disease between induced ITTP and real ITTP in humans due to non-inhibiting antibodies occurring in course of the real disease. I don't know if I really understand the question, but I would answer that indeed, if we induce TTP in the animals, we now inhibit ADAMTS13 function, and there's no clearance, at least not in the baboons and in the mouse models we developed. And it is true that in humans, clearance of ADAMTS13 by the antibodies is a very important mechanism. So I don't think that explains the severity, the difference in severity. I think the difference in severity is more related to the rodent models themselves. Okay, then what else? Would Estonia, would IVIG be effective to treat immune-mediated TTP? I'm not a medical doctor, but we discussed that with my colleagues different times, and apparently this is not an effective treatment for immune-mediated TTP. How do you explain that baboons develop hematologic abnormalities following anti-ADAMTS13 antibody injection without need for any other trigger? That's also a very good question. We do not really have an explanation for that. The only thing we can think of is that maybe baboons are more, how should I say, sensitive to endothelial activation or inflammation, and that is the reason why they give spontaneously these early stages of TTP, but the real reason we actually don't know. I think I answered them all then. I don't know if you followed, but okay.
I don't know if anyone wants to add any other question.
Can I ask it with the microphone?
Sure. Hi.
So, Johanna, can I follow up on this last question?
Yes. Sure.
That you don't need a trigger. Have you checked? I mean, on the ultra large von Willebrand multimers, the complement system can also be activated.
Mm-hmm.
Have you checked whether there could be a difference when ultra large multimers persist in baboons versus mice and rats? Whether this could be sort of mediated then finally the disease by complement activation?
That's a very good point, but unfortunately we did not do that. At that time we did not look into the complement system. No.
So thank you.